Hello everyone, good afternoon. Thank you very much for coming along to the Ensembl-84 release webinar. My name is Ben Moore and I am an outreach officer for Ensembl. So I'm part of a small team that's responsible for creating and developing workshops and holding them. And we also give webinars like this one. We're also in charge of the help desk system, so if you have any questions about Ensembl, you can contact the help desk, and one of the members of my team will get back to you about that. And we're also in charge of the social media, so if you follow us on Facebook or Twitter, you may have seen us there before. So just some background about the webinar interface. I had this slide up just before the introduction, in case any of you were early. But if you do have any questions that arise about the webinar today, then you can ask questions in the chat box in the webinar interface. So the webinar interface should look like something that you have on your screen here. And you can use this chat option here where my Ensembl colleagues will respond to you during the webinar. There isn't any threading in that chat box, so please use at your username, and then that's how we'll be able to thread the conversation so you can get answers to your questions that you have. And if there are any questions left at the end of the webinar, then we're going to try and visit some of those at the end after the presentation. So I want to start by giving you a quick background to Ensembl and genome browsers. So I'm sure many of you know that Ensembl is a genome browser, and it was first conceived of in the late 1990s, just prior to the publication of the first human genome. But the first genome actually to be sequenced was in 1977 by Fred Sanger and his team here in Cambridge in the United Kingdom. And it was at bacteriophage, and it was only 5 kilobases in size, which obviously isn't that big by today's sequencing standards. But that technology really laid the foundations for more and more complex organisms to have their genome sequenced until in 2004, the first draft of the human genome sequence was published. And this is 3 gigabases in size. So you can see how much the technology improved in just a few decades. Even since then, improvements in the technology of sequencing have led to the reduction of the cost and an increase of the speed of sequencing, which has led to more and more organisms to have their genome sequenced, but also the birth of projects that have aimed at sequencing multiple individuals within populations of a specific species, such as the 1000 Genomes Project you may have heard of before. So all of these sequencing efforts over the last 20 to 30 years, maybe 30 to 40 years, have led to the accumulation of a large amount of raw sequencing data, which without any annotation or context doesn't mean an awful lot. So this is where genome browsers such as Ensembl try and take this raw sequencing data, annotate it, put it into some context and make it accessible for scientists to make the most out of this sequencing data. So Ensembl does this in 4 main ways. So firstly, we have genomic assemblies. So for each of the species that we host in Ensembl, the genomic assembly is automatically annotated with our gene annotation pipeline with gene models. We also have a large amount of variation data. So we have both small and large scale sequence variants with the phenotype associations where that data is available. We also have a number of comparative genomics analyses. So we have whole genome alignments and gene trees in Ensembl, for example. And finally, we also have our regulatory build. So we use data from sources such as ENCODE, which is aimed at predicting the existence of potential regulatory features such as promoters, repressors, and enhancers. So to draw these 4 main branches of genome browsing together, Ensembl has a number of features. So for each of our species that I alluded to earlier, we have the gene build. And these are taken pairwise from species to species and compared to produce our gene trees. We also have our regulatory build, which is the prediction of the existence of regulatory features along the genomic assembly. And this draws in data from sources such as ENCODE. And this is something that we're going to pick up very much in the Ensembl-84 release. We also have a large amount of variation data, and we also have the variant effect predictor, which is a tool that we have in Ensembl that allows you to analyze the consequences of your own variation data. You're also able to upload your own data to Ensembl to view in the web interface alongside the existing Ensembl data. And we also have Biomarked, which is a data export tool that allows you to perform some quite complex data mining exercises and export data from Ensembl in that way. For all of these features, we have programmatic access, so you can access all of these features via both the Perl and the REST APIs. But the best thing about all of this is that it's completely open source, so all of the data, all of the coding, all of the scripts that we have that help Ensembl run and produce all of the data how you would see it in the web interface is available for you to take and use downstream in your own work, in your own research, how you see fit. So as I said, Ensembl has over 70 genomes, and Ensembl is primarily dedicated to vertebrate species. But we try and cover the broadest taxonomic space possible, while including important organisms that are important, for example, in human health, in agriculture, and in conservation, for example. So you can see here we have the 4 great apes, and we also have a number of other primates. We also have a large number of rodents and then other placental mammals. Moving up towards 2 o'clock on our dial, we also have some marsupials and a monotreme. Moving away from mammals, we have also a number of birds and fish, as well as reptiles and amphibians. And finally, we also have some non-vertebrate chordates and also some non-vertebrate model organisms. So we also have C. elegans, the fruit fly, Drosophila melanogaster, and Saccharomyces cerevisiae. And obviously these are not vertebrate organisms, but they are important biological model organisms, and they do form the basis of our comparative analyses, such as our gene trees. So we've also included these species in Ensembl as well. So Ensembl works in a release cycle, and this is the reason for our webinar today. So in December of last year, 2015, we had the Ensembl 83 release, and since then we've updated Ensembl with new genome assemblies. We've also updated software and gene sets, and we've incorporated a large amount of other data from updated databases as well. And we've packaged this all together and we released it all in Ensembl 84, which, as you know, was at the beginning of March this year. And this release cycle makes everything much more traceable. So if you've worked on a particular version of Ensembl before, perhaps you've worked on Ensembl a few years ago or a few months ago, then you can access the archives of Ensembl and view the data how you would have seen it at that time that you were working before. So now I want to move on to all of the new features that we have in Ensembl 84. Here I have just a few of the headlines. If you're more interested in all of the updates that we do have in Ensembl 84, then you can find all of the updates with a description using the URL at the bottom. But just to run through the main highlights of Ensembl 84, firstly we have the incorporation of BLUEPRINT epigenome data, and this is for humans. So in Ensembl 83 and in previous releases before that, we incorporated data from ENCODE and Roadmap Epigenomics in our regulatory build. But now we also have been able to incorporate BLUEPRINT data into our regulatory build, and this is something that we're very excited about, and I'm going to talk to you a bit about that in a moment. We also have a large amount of updated variation data. And moving on to variation, we also you're now able to calculate linkage disequilibrium in a pairwise manner between 2 variants of interest. You can also view the transcript haplotype view, which is a way to analyze the effect of 1,000 genomes variants. It's the way to analyze 1,000 genomes variants in a transcript dependent way. And I'm going to show you about the transcript haplotype view later in the webinar. Finally, the last thing I'm going to talk to you about in the webinar today is the Track Hub registry interface. And also importantly for mouse, we have updated the gene set to the gen code M9 annotation. And in zebrafish, we've also updated the gene set, which includes manually annotated Havana annotation. So firstly, I wanted to talk to you about the blueprint data. So you can find lots more information about the Blueprint project at the Blueprint homepage, which is linked via the URL here. But it's basically a large-scale project which was aimed at deciphering the epigenome of a number of different blood cells. So the projects mainly involved gaining data about the regulation of blood, the genomic regulation of blood cells using ChIP-seq and DNase hypersensitivity data. And in Ensembl-84, we are now including blueprint data for 20 independent cell types. So as I will show you later on in the webinar, these are not actually 20 different cells. They're 20 different cell types, so and they're also based on cell lineage and tissue source as well. So for some cell types, we also have data for cells derived from both umbilical cord blood and venous blood, for example. So as you can see here, this is a very rough example of the hematopoietic stem cell lineage. So you can see at the bottom here, we have a link to all of the cells that we have used for blueprint incorporation in Ensembl-84. But you can see here highlighted some of the cells that we do have in Ensembl-84 for the blueprint data. So we have erythroblasts, we have neutrophils, eosinophils, we also have monocytes and a number of macrophage lineages and differentiation paths. And we also have blueprint data for T and B lymphocytes. So now I want to show you how to access the blueprint data in Ensembl-84. So here is a screenshot taken from a region in detail page. So this is the normal interface that you will view when you go to any chromosomal location in Ensembl. So you can see here we have the chromosomal location, we have the chromosomal view, we have the 1 megabase overview of the region, and then we also have the region in detail view at the bottom. And you can see if you scroll down the page that this region in detail view by default has a regulatory track already added to the view, and this is a regulatory features for multi-cell. So you can see here using the regulation legend that in this region you can see a red promoter exists in this region. But this is a multi-cell feature, so this is the amalgamation of all of the different cell types that we have in Ensembl and a consensus as to where the regulatory feature lies within the genome. So if you scroll back up towards the top of the page, you can add blueprint data to this region in detail view by using the Configure This Page option. So I've highlighted the Configure This Page button on the left-hand side of the screen there. By clicking on the Configure This Page option, this will bring up a pop-up box where you can firstly add many different tracks. But if you click on the regulation link in the left-hand side of the pop-up box, this will bring up all of the different tracks that you can view that relate to regulation data. So you can see by default the multi-cell regulatory feature track was already added. But in this example, I'm also going to add regulatory features for EPCs in venous blood and also regulatory features for eosinophils in venous blood. And you can add these tracks just by clicking on what would be an empty box to the left-hand side of each of the cell types. You can add these tracks by clicking the tick in the top right-hand corner of the box. And this will bring up more tracks in your region in detail view. So where previously we had just the multi-cell regulatory feature track added to the region in detail view, we now also have the EPC and the eosinophil regulatory track added, and these are color coordinated respectively. So you can see for both of these cell types, we have an active promoter predicted in the same region. To view more data associated with Blueprint, you can again use the configure this page option to bring up the pop-up window, and this will and instead of clicking on regulatory features, I've instead clicked on histones and polymerases to view all of the data that we can find about histone modification and polymerase binding. So you can see here, this is quite a complicated view, but I'm going to walk you through it very slowly. So at the top here, we have all of the different cell types, and down the side, we have all different histone modifications, and further down, polymerase binding motifs. So to add data relating to the blueprint, you can hover over the cell type and select all, and this will highlight all of the data that is relevant for the specific cell type. As I've highlighted here in the EPCs, you can also change the style of the view by clicking on the box to choose whether you see the peaks of the data or the signal of the data. As you can see here now, I've turned the data on for both the EPCs and the eosinophils. All of the data that is relating to these cell types has been added, and I've set the style to both peak and signal. I can add these tracks by clicking in the tick in the top right-hand corner. So as you can see, even more data has now been added to our region in detail view. So you can see for the EPCs, there's a number of histone modifications that have been identified in this region in detail view, and the same for eosinophils. You can see histone modification, a number of different histone modifications that are annotated to this area of the genome. And so this is the peak, and this is the signal for each of these histone modifications, which have given rise to the annotation of the regulatory feature in each of the positions here for the cell types. So that brings us to the end of the blueprint data that we have incorporated in Ensembl-84. I now want to talk to you about some of the other updates that we have. So firstly, we have updated variation data. So variation data is brought in from a number of sources, and variants come in from a number of types. So firstly, we have small scale variations. So we have small insertions and deletions, or single nucleotide polymorphisms, or SNPs. But we also have large scale variation data, such as copy number variation, on large deletions, duplications, insertions, or translocations. And as I said, we get all of this data from a large number of sources. So these are publicly accessible databases that store variation data. So I just want to draw you to a number of these that are very important that we draw data that we've drawn for Ensembl-84. So dbSNP is a large variation database that includes variation data relating to single nucleotide polymorphisms. We also have incorporated new data from the GWAS catalog, also the online Mendelian inheritance in man, and also the DGV archive, which is a database that holds variation data relating to large-scale variants. To find all of the variation sources that we incorporate in Ensembl, you can use the link at the bottom of the page. So for Ensembl-84, we have updated a large amount of phenotype data for a number of species. So for humans, we have phenotype data updated from the GWAS catalog, from OMIM, ClinVar, UniProt, OrphanNet, the Cancer Gene Consensus, and GOA as well. We also have updated variation data for several species from GOA and OMIA, which is the Online Mendelian Inheritance in Animals. And we also have updated phenotype data for zebrafish from ZFin, rat from RGD, and mouse from IMPC and MGI. As I said, the DGV archive is a database of genomic variants that is a repository that provides archiving, accessioning, and distribution of genomic structural variants in all species. And in Ensembl-84, we have new and updated studies from the DGVA for human, mouse, zebrafish, macaque, cow, and dog. The last source of variation data updates for Ensembl-84 is dbSNP, where sequence variants from dbSNP 146 database have been incorporated in Ensembl-84 for human, cow, and dog. So as I mentioned in the headline slide, one of the new features of Ensembl-84 is that you can calculate the linkage disequilibrium for two variants of interest. So linkage disequilibrium is the association between alleles at different loci that is different from what would be expected based on their individual frequencies. So it's the measurement of the existence of two different alleles within the same sequencing data and assesses whether they come together more or less frequently than would be expected based on the frequencies within the population as a whole. So the R² and D' values are two common values that are used to summarize the linkage disequilibrium. And if you follow the updates of Ensembl regularly, you'll know that in Ensembl-83 we incorporated the Manhattan plot, where you could find both the R² and D' values for a variant of interest and the surrounding variants within a genomic location. But in Ensembl-84, you can now calculate the linkage disequilibrium between two specific variants that you're interested in. And I'm going to show you how to do that now. So by searching for a variant of interest in Ensembl, you can navigate to the variation page, and you can calculate the linkage disequilibrium to another variant by clicking on the linkage disequilibrium button on the left-hand side of the page. By clicking on linkage disequilibrium, you'll come to a page where you can see at the bottom there is lots of 1000 genomes data. There's lots of 1000 genomes project data, and there's also a box where you can insert your variant of interest. So by typing in the second variant of which you're interested in and pressing the compute button, Ensembl will calculate the R² and D' values for a number for all of the different subpopulations within the 1000 Genomes project. Sticking with variation data, one of the other things that we have updated in Ensembl-84 is the Transcript Haplotype view, which is a new view that allows you to explore observed transcript sequences that result from variants identified within the 1000 Genomes project. So you can view protein sequences, population frequencies, you can view protein consequences, population frequencies, and protein alignments of all the haplotypes for that particular transcript. So you can do this from any transcript page. So this is a typical view that you would see in Ensembl. We're currently viewing the BRCA2, one of the BRCA2 transcripts. And you can view the transcript haplotype view by clicking on the haplotypes link in the menu on the left-hand side of the page. By clicking on the haplotypes view, you'll be able to see all of the different consequences in terms of all of the different protein consequences for the transcript of variants that have been identified in the 1000 Genomes project. So if we take the top haplotype, for example, you can see that at position 2466, there's been a valine to alanine mutation, and this occurs at a frequency of 0.513 from the 1000 Genomes project, and this is being further split down for each of the different subpopulations within the 1000 Genomes project. So by clicking on the haplotype that you're specifically interested in, you can find more information. So you can find all of the broken down population frequencies. And so obviously the table that we saw on the previous slide contained all of the population frequencies for the population groups within the 1000 Genomes project. But this further breaks down each of the individual populations within the population group. So you can see the population frequencies in much more detail. You can also view, so one thing I forgot to mention was at the top of when you click on the transcript haplotype, you'll be given a menu where you can go where you can jump to population frequencies, which is what you're viewing here. But you can also jump to a number of other pieces of data associated with the haplotype. So you can jump to a line sequence, for example, which is here. So the aligned sequence shows you the reference protein sequence and the alternate protein sequence based on the variant that's been identified, and the alignment of the CDS sequence against the alternate alleles. So the last thing I want to talk to you about is the track hub registry. So again, in previous versions of Ensembl, you were able to directly add track hubs, but now with the birth of the track hub registry, you're now able to search the track hub registry for track hubs of interest and attach them directly to Ensembl. And you can do this in two ways. So firstly, you can search the track hub registry by clicking on the track hub link on the home page. But also from any region in detail view, you can add track hubs by clicking on the Add Your Data button that's just beneath the menu on the left-hand side of the page. By clicking on either of these buttons, you'll be taken to a pop-up window where you can search for track hubs that relate to specific species and specific assemblies. And by searching for a specific track hub, you can add them directly to the Ensembl and view them in the way that you would have done before. If you don't know, if you don't see the hub that you're interested in when you have searched it, you can manually attach any hub for which you know the URL as well. So that brings me to the end of the Ensembl 84 release webinar. I just wanted to wrap up with a few pieces of information before we get to some of the questions. So firstly, I wanted to let you know that you can actually host an Ensembl workshop. So as part of the outreach team, we can come and give an Ensembl course at your institution for free. And if you're interested in perhaps organizing an Ensembl course at your institute, and then you can email either the help desk or myself. And we generally run two types of courses, although we can tailor them specifically to your needs. So we can run either a short 1 to 2 day course that's just on the Ensembl browser, and that's aimed at wet lab scientists. Or we also run a 1 to 4 day API course that covers the Ensembl Perl API and is generally aimed at bioinformaticians. You can also learn more about Ensembl using the online tutorials that are linked via the URL. But you can also find lots of interesting information about using Ensembl through our recorded demonstrations on YouTube. And if there are any questions that perhaps relate specifically to your own work, or if you have even general questions about Ensembl or any questions about Ensembl, then you can email the help desk by emailing helpdesk@ensembl.org with your question, and one of the outreach team will get back to you there. So if you're interested in following us and learning perhaps about the updates for the latest Ensembl releases, and then you can follow us via Facebook and Twitter @Ensembl, but you can also subscribe to our blog, www.ensembl.info. And I'd just like to finish by acknowledging all of the Ensembl team that put in lots of hard work and effort in creating all of the all of the new features for the release and updating all of the data. All of their hard work and effort really does go into making the Ensembl releases possible. And obviously all of our funders, the Wellcome Trust, Ensembl, and the EU, and perhaps specifically for e84, the Blueprint project is very important